Great. Hi, I'm LESIA LIAO. I go to the Swarthmore College of Pennsylvania. This summer I had the opportunity and privilege to work in the lab of Dr. Quiñones-Jenab in the Department of Psychology at Hunter College, part of the SPUR program. This summer in our lab we studied the sex differences in anxiety-like behavior in response to a synthetic cannabinoid. So our lab studies anxiety. Just some facts and stats about anxiety. It is the most common mental illness in America. Also, which means it affects 40 million U.S. adults, which is about 18% of our entire population here. And it also costs the U.S. more than $42 billion a year, which is about a third of the mental health bill. And with all these numbers and its prevalence, this is why it's so important to do research on anxiety. For anxiety, women are 2 times as likely to express an anxiety disorder. They also occur earlier in women than in men. People who suffer from anxiety are also more likely to abuse illicit drugs such as cannabinoids. Cannabinoids are commonly associated with cannabis or marijuana. Cannabinoids, they act on cannabinoid receptors in the cell and have a two-way effect on anxiety. So high doses can increase anxiety while low doses can actually decrease anxiety, depending on what dose. So with the rise of cannabis use and the number of people being diagnosed with anxiety, there has been an increasing amount of research done on the two. However, the mechanisms and interactions and relationships between anxiety, biological sex, and addiction have not been well understood or studied. Most of the studies on drugs and addiction look only at males, even though the number of females using and abusing drugs have been steadily increasing. Though it makes sense at first when you do research on a new drug, new research, to do it on males because it's a control. But with all the rise in interest in women in STEM fields and studies, we wonder, why isn't there more research done on females? Currently, there's not enough scientific literature on females in rodent models, and humans, and so this is why it's especially important to study, because as we learned earlier, anxiety affects women 2 times more than men. And this is why our lab decided to study sex differences, anxiety-like behavior, in response to cannabinoids. Like many psych labs, we chose to use the classic rat model to explore the relationships between the brain and the behavior. So in the rats, we can study their behavior, and also we can look at their biochemical changes in the brain after they behave and we have to sacrifice them. So rats are interesting because they have an eternal conflict of interest. So part of them want to explore, and another part of them wants to stay safe and hide in the shadows. Scientists have been able to exploit this approach avoidance conflict in the elevated plus maze. So the elevated plus maze is a test for anxiety. What you do, it's made of 3 parts. You have closed arms, open arms in the intersection, and what you do, you place the rat in the middle of the maze, and you allow them to explore for a set amount of time, and you record its behavior, where it goes, and how long they're there for. To break it down, we have the closed arms. So closed arms are indicative of anxiety-like behavior. So rats are prey animals, and they want to avoid all the predators that want to eat them. So naturally they want to go to a covered space which is enclosed if they're anxious. There's also the open arms, and the open arms is more for the other side of the coin, so the exploratory behavior in the rats. And then last but not least, we have the intersection, which is what we want to study. So the intersection, it's neither open nor closed. So it's like kind of in between, and previous literature suggests that it represents risk assessment, which is a measure of anxiety. So risk assessment kind of falls under the big umbrella of anxiety, and it's the specific measure that we're looking at in our study. So we hypothesize that female rats will show more risk assessment behavior than males in both a cannabinoid control and cannabinoid treatment. And this could be characterized by spending more time in intersection, because as we learned, so more time in intersection is indicative of risk assessment behavior. So to study this, we looked at male and female rats. We injected them with a single exposure of a moderate dose of CP55940. Now CP55940 is a synthetic cannabinoid, and it acts like THC, and THC is the main component of cannabis. So, and it has similar receptor activity and behavior effects of THC. So we're hoping that this can parallel with anxiety sufferers using cannabis to treat their anxiety. After we injected it with the synthetic cannabinoid, we tested them on the elevated plus maze for 10 minutes. When all the testing was done, we analyzed all the data with statistical analysis. We used a two-way ANOVA test and independent t-test afterward. So, what we found was in the ANOVA test, we found there was an overall sex effect. So, in all our data, there was a sex effect on the time spent at the intersection, but there was no effect on dose nor interaction between dose and sex. But once we found out that there was an overall sex effect, we did an independent t-test to see where was this effect. Was it certain to a specific group? But we saw that in both the control and the drug group, there was a non-significant effect for sex. But if you look in the graph, you can see that females spend more time at the intersection for both conditions. So more time at intersection, more risk behavior. And also in the males, which is the gray on the right, with the drug, the male showed a slight decrease in time spent at the intersection. We tried to take a closer look at this. So we did a test on the effective dose within the sexes. So we found out that in both females and males, the treatment, the synthetic cannabinoid, had no effect on the time spent at intersection. So basically saying that the synthetic cannabinoid in this dose didn't impact their time at the intersection at this particular dose. So with this knowledge and analysis, we can see that sex does affect time spent at the intersection. With the ANOVA test, the females did spend more time than the males regardless of the treatment. And so regardless of the treatment, so with that we can test at different doses and see where does it change, maybe at a higher or a lower dose, because this was a moderate dose. So it was pretty variable where it could have gone here. Also emphasizing that spending more time at the intersection of the elevated plus maze is a measure of risk assessment. So yes, this does support our hypothesis that there was a sex difference in risk assessment in the rats. Some limitations in our experiment were that the tracking software that we used didn't allow us to study other possible measures of risk assessment. So besides time at the intersection, you could study risk assessment with number of head dips, grooming, and the feces count, and the locations of that. For future research, we're currently in the process of performing Western blots on 5 brain areas that are critical to memory, decision-making, voluntary movement, pleasure, and emotion. And we're hoping that by doing this, we can find a relationship between changes in the protein levels in the brain and risk assessment to see if there's differences between the sexes on a biochemical level. So yeah, thank you Hunter College Psych Lab, thank you for all my lab mates and mentors, thank you SPUR program, NIH, and the Leadership Alliance for giving us this opportunity to share our research.